Share screen. Okay. Yes. Now, in realistic situation, we take a one plant of yellow and round seed, and we take a green and wrinkled seed plant, and we cross. We get only one plant. So we can't predict that, but we can say that this plant maybe outcome is outcome maybe uh, out of this 16 combination to become a green and wrinkled, right? So become a green and wrinkled, this one. So if chance to become green and wrinkled 1 in 16, because there are 16 combinations, out of 16 only one combination coming green and wrinkled, then we can say, okay, do 16 times. If you do 16 times, you may have a chance to get one plant green in wrinkle. So this the Punnett square is something we make to predict the chances. So we don't it doesn't get everything. Like we don't get, for example, if Mendel did this experiment 16 times, so he may get a 9331 ratio, but to increase the probability, chances, we can't do 16 times. The simple example, you toss a coin in front of you. You have a chance to get head or tail. But first prediction, someone can say you get a head. It's a prediction, head or tail. So if you do first time you get a head. Second time also you get head. That means it is not 50%. If you do two times, one can say you get first time head, maybe second time tail, or first time tail, second time head, because chances are 50%. But it can't happen like that. You get a head, second time you get it, even third time you can get a head. But if you are done 100 times, then your chances are very close to 50-50. Now Mendel did this experiment 16 times, 32 times, like 46, 64 times, like that. He did many times, and he observed the ratios are 9331, like in the first experiment. So what we did here, all the possible combinations. So during gamete formation, allele separate, then this allele has a probability or chance to combine with each other. So these chances, probabilities, we can write on this Punnett square all the possible combinations.
Uh, so during the formation of gametes, does DNA replication occurs?
DNA
Replication.
during formation of gametes, during DNA replication. Yes, I think you can check the meiosis. What happened actually in human cell, there are 46 chromosomes. There are 46 chromosomes, and these 46 chromosomes, okay, it's something like this, right? How to say? Okay, I will briefly explain it. Now, let's take a good example of male. The male testis is the place gamete produced. Now, when you take the testis gamete productions, the sperm cell production is a continuous process. That means every day sperm cell produce, and there are about 50 million, more than more than 50 million, I think close to 500 million, 50 to 500 million sperm cell can move in one ejection. That means millions of millions cells produce in the testis. To produce that much of cells, the testis has cells. These cells are the germinal cells. These germinal cells undergoing mitosis. So germinal cells undergoing mitosis, and they always keep this number to a certain level. Let's say the germinal cells, I mean, let's take one example like 1,000, good example. They always keep this 1,000 because they continuously produce cells to maintain 1,000. And some of the cells, they are moving to the next level, they send to form sperm cells. So they continuously make cells and keep 1,000, and when they produce more cells, they send to produce sperm cells. Now these cells, when they send to form sperm cells, they are 2n. Now this 2n cell has 46. Now this 46 replicate to make 92. Now this is DNA replication take place here. So it become now 92. And 92 undergoing meiosis. During meiosis, again it formed 42 and 42. This is the meiosis now. So here DNA replication take place. But you know why we call meiosis? Only it separate the homologous chromosomes. So one chromosome of homologous pair is going to the other side. The next one is going to the other side. That's why it's 42. Number wise 42, but the quality wise they are half. That's why we call meiosis 1 is the real reduction, but not the number, but the quality. Then after that, this again undergoing second meiosis, 23, 23, 23, 23. Yes, DNA replication take place at this place. And why did you ask the DNA replication take place?
So if you take the capital Y and capital R combining with simple Y and simple R, we get 4 gametes of 2 capital Y and S.
Okay, again capital YN.
Capital Y and if you take these capital Y and simple Y,
Capital
the,
Y
cap
and simple Y, yes.
the capital Y should replicate and simple Y should replicate and they undergo meiosis. No, sir?
Yes, yes, because the homologous pairs, if you take one pair of chromosome is like this, other pair is like this, and what will happen during replication, after replication, it become like this, one and the next one, right? So then this one and this one identical, this one identical. So they are identical, right? So if in case we have a capital Y here, and in this case simple Y here. Then we get a capital Y, simple Y, simple Y.
Got it.
And then meiosis, what happened? They are separated. This goes to one gamete, this goes to one gamete. But don't take all these crossing overs and many other things which are happening, which does not actually included in Mendel genetics, because Mendel was so lucky that when he took this plant, when he took these alleles, they actually completely work independently. No other theories applied, so he got luckily these ratios. Nothing other phenomenon happened. So that's also one good thing, that it's really lucky that he when he select these plant and characters, they actually really help him to find this uh, the ratios and the laws. Okay, now he could repeat this experiment using again 600-700 plants, and he observed 9331. Then he can again repeat 9331. Then he came up with the second law. So we can say what has happened in this case. First one, right? The alleles separate and pair up to make the gamete. So we don't want to reinvent. We just say, by looking at the pictures, the alleles separate and pair up. So if you take example, the red one, alleles separate and then pair up, right? So we write it, what has happened. And pair off to form gametes. And next thing, once this gamete form, the rounded, orange color round shaped gametes in this Punnett square. So what we did, we tried to find all the possible combination. Then the gamete assort independently, right? So if we take this one and this one, combination is called assort. The gamete assort independently. Independently. Then the gamete assort independently. So this is called law of independent assortment. This is a Mendel second law, law of independent assortment. So it came up after this dihybrid cross.
What is the thoughts?
Assort is, I think we can write a combine. So this the gamete combine independently. As a result of that, at the end, that the alleles, the genes are actually the gamete assortment mean genes are combining, because here when we write capital Y, capital R is the gamete. This is capital Y, capital R is a gamete. When the gamete combine, it actually combine the genes, because gamete combines mean genes combine. So the meaning is same for both the gamete assort or genes assort independently or allele assort independently. Right? So we can say gametes or genes. Gametes or genes assort or combine independently. Alleles separate and pair up to form gametes. Then gametes or genes combine independently, assort independently. I think this is almost end of Mendel genetic story. Mendel, Mendel genetic is still valid, and some of the characters which are inherit according to the Mendel's law. Not everything, but still some of the characters inherit according to the law of Mendel. So, what is important? There are some Mendel law calculations. So, based on these two laws, the probabilities, we have to practice some of the questions. But we can do after we finished the complete Mendel lesson and understanding the probabilities, then we can practice these questions, because typical genetic questions, they were asked what is Mendel first law, what is Mendel second law. Instead they are asking some practical questions. There is animal that has this allele and then combine, what is the probability of getting this combination, like this, like a mathematical questions are asked in the exam. uh, to finish Mendel genetics, we try to learn a few things. Why Mendel was successful in his experiment? Because Mendel used large sample size. The one success is large sample size. Large sample size, and many times repeated the experiment. Now, why this is important? As I mentioned you, the coin, tossing a coin. If we have done it only one or two or three times, we never get some ratios. But if we done many times, we get ratios. We get fixed ratios. Right? Large sample size and many times repeat help him to get a fixed ratios. Then he was successful in his experiment. He could predict now, because he could predict what is the outcome. So this able to predict, because the ratios are constant, then he can predict, okay, do a 100 experiment, you get ratio like 9331. You get 3:1 ratios. So that is one uh, thing. And then he also followed the F1, F2, like this many generations, did for many generations. It just not ended up in one generation F1, but he did for many generations. And then analysis and records. So frequent analysis and record keeping is also one of the thing he did for his success. On the other hand, he also gets some kind of luck for his success. That is the pea plant has this the self pollination and cross pollination. Because earlier we learned that mostly they are having the self pollination, but we can also do cross pollination, and generation time shorter, because he could quickly do a breeding experiment and then he can get the result. If he wait for years, then it doesn't work. So this is also one thing. And I think last, uh, in terms of modern genetics, they are located on separate chromosomes. The characters are located in different chromosome. So if it is located on the same chromosome, they are not separating, because when we say all alleles separate and independently assort all these things, if they are separate, but if they are in the same chromosome like this, capital Y, simple R, capital R, simple R, like this, they don't separate. They always go as a one chromosome to the gamete. So these are the few things we can consider: genetic perspective, the plant, and as a person, uh, what he did all contributed for his success. Okay, now we will try to understand these probabilities. Okay, a basic the probability is uh, it's a chance. So what is the chance or likely to occur, one event likely to occur out of number of possible outcome? One event likely to occur out of several uh, events. One event likely to occur out of several events. I think the simple one is the dice. So if we take a dice, so it has six sides, number of events. The probability is getting one event, number of events, number of total events. Let's say probability of getting 1, 2, and 6. So it is usually 1 out of 6. Getting a number, predicted number, 6 times we roll the dice, then chances are 1 to come up the desired number. That's a basic probabilities, right? So that's why we use Punnett square 16 total number of events, possible events, and getting a green wrinkle is 1. So this is 1 in 16, and this is 3 in 16. This is 3 in 16. This is 9 in 16. So because there are 9 probabilities getting round and yellow one. Okay. Right now, we are coming to another concept of probability. Now, to understand this, you think this example. This is a coin, and this is another coin. So we have two coins. Now, if we have two coins, and we toss the two coins at the same time, what is the probability that we get the head on both coins? Right? So toss the both coins at same time. What is the probability to get the same outcome? Example, head on both. Now you see some differences because one coin has chance to get a head or tail half, right? So getting head or tail, the probability is half. This coin getting head or tail, probability is half. But now there are two independent coins because one coin is doing independently, other coin is doing independently. Then how we can predict that both get the same thing at the time? For this kind of a thing, we have a multiplication rule. Multiplication rule. So this case, what we do, the probability, that means this one, right? The first one we learn what is the probability of the one event multiplied by probability of the second event. In this example, half multiplied by half, answer is this. Now answer for this question is this one. So you had to use probability of multiplication rule. Now why this is important for our genetics? So you can understand, if this is the egg cell and this is a sperm cell, so we have to use this kind of a multiplication rules to predict the probabilities. So we have to use some instances this multiplication rule. Now we take one example how nicely this probability of multiplication applying in genetics. Okay, we haven't learned yet, but we have to learn in the future. But we learned the simple thing. 23 human chromosomes, 21 and 1, like this. So there exists 21 autosomal chromosome, and this is a sex chromosome. Now sex chromosome is labeled as X and Y. Now what is the function of sex chromosome? They determine gender, that is male or female. Now male has XY, female has XX chromosome. Now if we try to find the probability of getting male or female, the man has XY and the woman has XX. So if they want to make a children, what we do, we make the Punnett square. Man has the gamete X and Y. The female has X and X. Because our rule, this one is haploid condition. When the gametes form, alleles separate into each gamete and they have a chance independently assort the second load. So we use the first and second load, alleles separate first load, and they are in the gametes now. And the gametes form independently assort second. So then XX, XY, XX, XY. So 50% and 50%. So chances are 50-50. So it's like again the coin, we need one half like this. Now, if we try to use this multiplication probability rule, we do this example. The first, the first child is male. Right? So our prediction is the probability is half. To getting a male child is half. Now, what is the probability second child is also become a male?
Half,
Half?
half into half. Oh,
Yeah, I mean, second child male or female, it's half. But second child is male is we have to use the multiplication probability rule. It's this because it's a first event is happen and the second event is independent now. Again, it's independent. So then you have to use the multiplication rule to find the second child become a male. I think many genetic questions. This is one place they apply this multiplication rule to find the male, female, second child or third child like that. They can use the multiplication rule. Okay, right. The last one now. This is the basic probability rule. Second one is a multiplication probability rules. There is another one that is uh, the adding rules. The probability addition rule. Now, how we can understand this? We take this example. So there is a bag, and this bag has marble with red color 1, 2, 3, and it has a marble with blue color 1, 2, 3, 4, and it has a marble with yellow color 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 3 red, 4 blue, and 3 yellow. Okay, now what is a probability? Right here, what is the probability of getting red and yellow marble at the same time? So we put a hand into the bag